Hi everyone! In the next two videos in our series, we're going to tackle horizontal gene transfer. And that's the transfer of DNA from one bacteria to another via a non-parental source. And this will make more sense once we've gone through the different processes. In the first video, we're going to look only at conjugation. And in the next video, we'll look at both transformation and transduction. So let's get started. First up, we have conjugation. Conjugation requires direct contact, and that contact is between two bacterial cells. It involves the transfer of a plasmid, a small circular piece of DNA, and that plasmid is going to be transferred from one cell to the other cell. And it's going to be transferred through what we call a sex pilus. This is basically a hollow tube through which DNA can be transferred. Up here we have what we call an F-positive cell. That means a cell that's able to make a sex pilus. And it is going to create and extend a pilus to come into contact with another cell. We call this other cell the F- cell. And that F- cell is going to receive the plasmid. As you can imagine, this is a really long way to try to transfer some DNA. That's a long tube to transfer DNA through. So once the sex pilus has been created, it actually retracts and pulls the bacteria really close together and forms what we call a mating bridge, which is what you see here. So we have one bacteria here, and they are super close together by that tiny mating bridge between them. And that's where the plasmid DNA is going to be transferred from one cell to the next. Before we get into any specifics about how this works, let's erase a little bit and let's make sure we understand some definitions. First, we need to understand what it means for a cell to be F plus or F minus. Now, I already said an F plus cell can make the sex pilus, and F actually stands for fertility. So an F plus cell has what we call the fertility factor or the fertility plasmid, and that plasmid contains the genes to make a pilus. So our F-positive cell is going to be our donor cell. It is going to donate plasmid DNA to our F- cell. Our F- cell does not have that plasmid, and so it will be our recipient cell. It is going to receive DNA from the donor cell. Now, bacteria are pretty promiscuous. They don't really care if the bacteria that they're reaching out to and undergoing conjugation with are of the same species or not. So this can be a transfer between any bacterial species. Okay, let's look at how this works. Right up here we have our F-positive or F plus donor cell. Inside it, we see the normal chromosome of the bacteria and the F factor plasmid. And again, that plasmid just contains genes to make a pilus. Over here is our F- recipient cell. It only has a chromosome. It does not have the genes on the plasmid to make a pilus. Our donor cell makes these sex pili, reaches out and touches the recipient cell. And then those pull the cells together. Now that our cells here are in close proximity, our mating bridge is formed. So there's a little gap here between our cells where they're connected to each other. And our plasmid, which has its own origin of replication, is going to start being replicated. Now, this type of replication that plasmids do is called rolling ball replication. And it is different from the DNA replication that we've talked about before for bacterial chromosomal DNA. In rolling ball replication, we start with our circular double-stranded plasmid like this. And once a little replication bubble is formed, I'm going to attempt to draw that up here. Bubble right there. There is going to be a nick induced, a little cut induced in our newly replicating plasmid. That is going to allow for our plasmid replication to occur like so in a linear fashion. And this newly synthesized piece of single-stranded DNA is actually what's going to be passed through our mating bridge right here. So we have our rolling ball replication occurring, and that newly synthesized strand is being passed into our recipient cell. Once in the recipient cell, the replication of that plasmid will be completed and it will be circularized. And now both of our cells, our original donor cell is still F positive, it's still F plus, because it replicated and shared a copy of this plasmid. And our F minus cell is now F plus also. And now it has the ability to make a sex pilus and carry out conjugation with any cell it comes into contact with. Now, most often plasmids stay as plasmids. They stay as circular extrachromosomal pieces of DNA in the cell. But occasionally, those plasmids will actually integrate with the bacterial chromosome. And when this happens, we call it a recombination event because we are combining the plasmid with the chromosomal DNA. Look up here at the top where we have our F plus cell. It has the F plasmid in purple and its bacterial chromosome in this sort of green color. For a recombination event to occur, there have to be areas of sequence homology on the plasmid and on the chromosomal DNA. And this allows for the two pieces of DNA to kind of intermingle. And what will happen is now we have our plasmid that has been inserted right here in purple into our chromosomal DNA, right? This we refer to as HFR, which is not the best name, and it stands for high frequency recombination. HFR means high frequency recombination. When an F plus cell undergoes this recombination, it is now called an HFR cell. Now, one last thing I want to point out about this. The arrow here in our plasmid, that represents the origin of replication, or the ORI. You'll notice that here in our HFR cell, that origin of replication is now in the middle of where our plasmid has integrated. So when replication begins at the origin of this plasmid, it is only going to replicate part of that plasmid DNA, and then it will continue along replicating the chromosomal DNA as well. Now, an HFR cell can still transfer genetic material to another cell. If that plasmid that was integrated has the F factor in it, it can form a sex pilus and reach out to another F minus cell. Just like with regular old conjugation, the two cells will be pulled together and a mating bridge will form. But now, instead of just replicating the plasmid, you can see that part of the chromosomal DNA is being transferred to the recipient cell also. So in HFR transfer, part of the plasmid plus some chromosomal DNA get transferred to the recipient cell, or to our F minus cell. On our diagram over here, you can see that purple part of the plasmid, just a little tiny bit of the plasmid has been transferred, along with several chromosomal genes that are being copied from our donor cell chromosome. We can monitor how long conjugation has gone on by knowing the distance of these genes from each other. So we can take a look at the DNA that's transferred to our recipient cell and figure out how long conjugation has lasted. If conjugation lasts about a minute, we'll only get a few genes. If it lasts 9 minutes, we'll get maybe another gene. 11 minutes, we'll get this number. 18 minutes, we'll get a lot more genes. Now, in the last slide, recall that I told you that the origin of replication of our plasmid is now in the middle because of the way it integrated. And that means that only a portion of the plasmid genes gets transferred. And this means that those genes for a pilus are not the ones getting transferred. And thus, at the end of conjugation between an HFR and an F minus cell, our HFR cell stays HFR. It's only giving a copy of its DNA away. And our F minus cell stays F minus. It does get new DNA. It receives some chromosomal DNA and some plasmid DNA, but it does not get the genes for pilus. Now, I know you've learned about plasmids in your intro biology classes, and you already know that they are very small, self-replicating circular pieces of DNA. In fact, we just went over how they replicate by that rolling ball replication rather than the usual chromosomal DNA replication. They are very small. They're anywhere between 1 and 5% of the size of a bacterial chromosome. So they don't have a whole lot of genes in them, but they can pack a lot of important information into each plasmid. When it comes to human health, often plasmids code for proteins that enhance their pathogenicity or make them more disease-causing. So let's look at a couple of examples of what plasmids can carry. One of the most important things that bacteria can transfer by conjugation are genes for drug resistance, so antibiotic resistant genes. And again, those can be passed from one species to another, not just within the same species. Another important thing is toxin production, or the production of molecules that are going to cause disease. A prime example of a toxin is produced by Staph aureus, and it's called exfoliative toxin. And what it does in young infants who are infected with Staph aureus is it causes their skin to peel off in sheets, like you can see in the infant pictured here. So it causes like a sloughing of the skin in young children. And the name of the disease that this causes is staphylococcal scalded skin syndrome, or SSSS. Additionally, genes for capsule can be passed by conjugation, and remember, capsules are useful at immune evasion and also at protecting bacteria from the external environment from things like drying. Despite being very small, like we see in figure B down here, plasmids, as I mentioned earlier, can contain a lot of information. And they don't have to contain just one type of gene. They don't have to have just pilus genes or just antibiotic resistance genes. They can include a variety of gene types on each plasmid. If a plasmid contains genes for antibiotic resistance, we call it an R plasmid. F plasmid, or F factor meant fertility plasmid, fertility factor. R plasmid means resistance because it carries out antibiotic resistance. In the plasmid shown here, we have genes that provide resistance to sulfonamide antibiotics, chloramphenicol, streptomycin, and tetracycline. So 4 different types of antibiotics just on this one plasmid. In addition, this plasmid also contains resistance to mercury, to toxic mercury compounds, while also having pilus and conjugation proteins as well. So a lot of different things on this one plasmid. And imagine if a bacteria suddenly picked up this plasmid from another bacteria or another species, and it went from no antibiotic resistance to all of a sudden having 4 different types of antibiotic resistance. Okay, time for a quick rapid review. Conjugation requires direct contact between 2 bacterial cells. An F plus donor gives some DNA to an F minus recipient, and our F minus recipient then becomes F positive itself. That F factor, that plasmid can integrate, which takes our F plus cell and converts it into an HFR cell. Our HFR cell can still share that DNA with another cell, but our F minus cell stays F minus because it only gets a portion of the plasmid genes. Plasmids themselves can contain toxins, genes for antibiotic resistance, and genes to make a pilus.